Nils.
Yeah, and I'm obviously Kira. And we represent the iGEM team from Uppsala of 2014. And for those of you who don't know, iGEM is an international contest in synthetic biology. And traditionally, what a team does is that they focus on an issue that they want to resolve with synthetic biology. But we thought differently. We wanted to solve a lot of problems with our project. We wanted to have a wide application for our system. So we wanted to combat bad bacteria in different environments and in different systems. But the problem with doing a very wide application is, how do we get specificity? How do we make sure that we will want to get rid of bacteria in like water? How do we not harm the ecosystem? And if we want to get rid of a bacteria in the body, how do we not harm the bacterial flora that we want to have?
So you're saying it's a bit like trying to kill one person with an atom bomb?
It's not very effective.
So our solution to this is the bacticide, the future of microbial combat.
But Nils, what is a bacticide?
Well, a bacticide is an engineered bacteria that is able to sense a target, then move towards the target. When it gets close, it starts to produce this weapon against its target. And when it's reached high concentrations enough, kaboom! Victory. So, as you might imagine, this, as Kira said, this will have a wide range of applications, but we couldn't possibly do them all in one summer. So we decided to focus on one, pathogenic bacteria in the gut.
But now that we have such a wide system, why would we choose to focus on the gut? Well, we had a lot of inspiration considering this. Last year's team from Uppsala focused on working with probiotics in the gut, and we thought maybe we could further explore what we could do with synthetic biology in the gut flora. And we also wanted to contribute to the problem, not contribute to the problem, to the solution of the problem of antibiotic resistance, which is a very big issue right now. And we talked to a senior specialist at the University Hospital, and he gave us a really, really long list of gut bacteria that he wants us to possibly get rid of. But the problem is, we want specificity, so we want to pick one bacteria. And how do we choose? Because the list was like this long.
Yeah, so that sounds like way too much. So what we did was that we set up a set of requirements. If we can fulfill these requirements, we can make an effective bacticide. So our first requirement was that we wanted an individual quorum sensing language.
So you mean like a language that only one strain of bacteria can understand?
Exactly. And also we wanted it to have a sensitive, a toxin that is sensitive only against that bacteria and nothing else. So, considering these factors, we decided to choose Yersinia enterocolitica. So, Yersinia enterocolitica is a gut pathogen that is related to Yersinia pestis, the cause of the bubonic plague. But don't worry, Yersinia enterocolitica does not cause the plague. Yersinia enterocolitica causes stomach flu and is commonly found in food poisoning, so like contaminated meat or infected lettuce. So, Mexican salad, be aware.
And this is the plan that we devised. We're going to use specific sensing, targeting, and killing. And this is also how we chose to divide the lab work during the summer, because to be effective, so we worked in 3 modules. And the first module we're going to talk about is the sensing module. And like we said, it's based on quorum sensing, which, as you may know, is how bacteria talk to each other and communicate through signaling molecules. And what we did was hijack Yersinia's very own quorum sensing system to get the specificity. So we had the GEM system with the N-box and GEN-R. And as you know, a quorum sensing system is 2 components. One produces the signaling molecules and one senses them and reacts to them. So the system that we have here allows us to control gene expression depending on how close we are to a colony of Yersinia. Because when we are far away, there won't be any signaling molecules, and so the activator, the NR, will bind to the N-box and give us a lot of expression. And when we are close to a colony of Yersinia, there will be a lot of sensing molecules, and we won't get any activation from the NR, and we'll get very little of the gene that we want to control. So that's officially gene expression control.
Wow, but Kira, how do we test this system?
Oh, I'm so glad you asked. What we did was, the gene that is controlled by the GEN system is here a blue fluorescence protein, which means that when we are close to Yersinia, we won't glow very much, but when we are far away, it'll glow a lot. And we tested this system by fuxing the system. because the FACS is a system that measures the fluorescence of this blue fluorescence protein. And as you can see, no gen R low expression, gen R high expression, and coordinate sensing molecule low expression. And we actually got results that proved that we were able to control the gene expression based on the gen box and gen R and coordinate sensing molecules.
Wow, amazing. But how does this on and off thing really help our system? Well, it's actually really important in our targeting module. In our targeting module, much like each cell has their own way of talk, they also have their own way of move. So, to control the movement in an E. coli, you can control a gene called KZ. So, if you produce a lot of this protein called KZ, the cell will move in large jumps, moving in random directions. But if you have very small amounts or nothing, the cell will just run around in circles like a dog chasing its tail. So, now that we've seen this, we want to be able to control this KZ. But to do this, we wanted to make sure that the internal expression of KZ in the cell doesn't interfere. So, we used a mutant of E. coli, which had knocked out the KZ gene. So that's a mutant that can only move in circles. So, are we actually able to restore the chemotaxis by just reintroducing the KZ gene? Well, yes we are. So here are some of our results. And what you can see is, up here is a negative control. That's the mutant I talked about that cannot move. And here you got two positive controls that move regularly, and here you got something that did not work, but look here. We are actually able to restore chemotaxis or the movement of the cell by just introducing this KZ gene. So we actually managed to restore the movement of the cell.
And this sounds like it would work very well with this gene expression control on and off.
Yeah, well, let's just combine them.
Sounds great. So what we did is, instead of blue fluorescent protein, we have KZ controlled by the gen box, meaning that when we are far away from Yersinia, we'll move a lot until we reach Yersinia, when we'll stop and simply chase our tails like a dog. Yeah, Niels, you might wonder, why is this important, being able to stop and chase our tails?
Yeah.
Well, I'm going to tell you, it's because the killing system needs it. The killing system is specific in two ways. For one thing, the toxin, the bacteriocin that we choose, is only produced when we are not moving in far steps, only when we're close to Yersinia and spinning around. For another thing, the toxin we produce is a colicin that's specific to Yersinia enterocolitica. It can only kill Yersinia enterocolitica. So that's a double specificity.
But wait. You said when we're in the sensing system, when we are far away, we produce a lot. But when we are close, we only produce a little. This sounds like the opposite of what we want in the killing system.
It's okay, Niels, I thought about that. See, instead of controlling the production of bacteriocin with the gen box, we control the production of an inhibitory molecule, a silencing RNA called Spot42. This means when we are far away from Yersinia, we will get a lot of Spot42, which will inhibit the production of a bacteriocin. Not very much bacteriocin when we are far away. Once we get close, we stop, and we produce a lot less of the inhibitory RNA, and we get a lot more of our bacteriocin. So you might also wonder, how did we test this?
Well, firstly, we wanted to know, could we actually produce this toxin? And yes, we could. So this is what's called an Sds-Page, and it basically shows us what's the length of this thing that we're trying to purify. So we know that our toxin is 50 kilodaltons. And as you can see here, I hope most of you can see it, is that there is a clear band here. And in our negative controls, we got no bands here. And this means that we're actually able to produce our toxin in our bacteria. And further on, we didn't settle with just producing a toxin, we wanted to know that it worked. So we actually went to Livsmedelsverket, the food authorities here in Sweden, and we were actually able to test our toxin against the real deal, against the real Yersinia enterocolitica. So what you see here is a plate where we just have grown commonly Yersinia enterocolitica, just living happily. And over here, we have the plate where we have added our toxin. And as you can see, there's a large difference, and our toxin actually seems to work against the real deal. And now we are actually doing experiments to see, is our toxin specific? Are we able to kill only Yersinia and nothing else? And the results are looking promising. So, the entire system of our bacteria would look something like this, with two modules, a tracking module or a tracking mode and an attack mode. So when we're in the tracking mode, we produce this GNR, the blue fluffy cloud from 4, and that amplifies the production of key Z, making us move in large steps. But we also produce this silencing part that makes us not produce the toxin, so we are sure that we do not put toxin on good bacteria.
Yeah, and in the attack system, as you can see, once we got close enough to Yersinia here, we stop moving, start spinning around, that's not, and we start to produce the toxin that will kill the Yersinia. That's pretty brilliant, huh?
Yeah, it's great.
I also heard though that this summer you did quite a bit of modeling.
Yeah, me and the boys, we went to Paris, we went to the catwalk.
I meant the other kind of modeling.
Oh, the one with the computers and math and stuff.
And coffee and
Oh, yeah, I get it. Well, so we actually wanted to test our system and see, does this work in the human body? But instead of taking the bacteria and giving it to someone and then sticking a camera down their throat, we thought, let's simulate it in computer. It's a bit more nice. So, our first model was, we call the cell-to-cell model. So this shows interactions between cells. So this is kind of what you would expect when you're looking in a microscope. So, in red here, we have the Yersinia, the bad bacteria. And in blue here, you can see small tiny dots. These are the tracking bacter cells. And when they get close, they will turn cyan and start killing. There is a video to this, but it's a bit hard to do videos when you're doing a PDF presentation. So instead, we have the clip on our YouTube page. So make sure to check out YouTube iGEM Uppsala and see our nice video. It's quite funny. It's also called Bactervid 1942. You can guess where that comes from. And also, we didn't settle there because we wanted to know, can our system actually work like in broad scale? Because that was only a small fraction of the entire intestine. So we set up all the Yersinias in our stomach and simulated what would happen to them. So this is the important graph basically. So what we can see is that they are decreasing in this kind of exponentially manner. So we're actually able to kill Yersinia, perhaps not all, but a large chunk of them at least.
Yeah, so the accomplishments that we have been able to get this summer is, for one thing, with the bactericide, we managed to prove that.
We can produce this toxin in E. coli.
And?
That we can actually kill Yersinia enterocolitica, the real pathogen bacteria.
And?
We can do, the killing is really specific and doesn't kill the good stuff. And with our targeting module, we were able to prove that.
We can restore chemotaxis. And in the modeling, we found that.
We were actually able to produce a successful intuitive cell interaction model.
And?
We were also able to produce an efficiency model of our populations. And in the sensing module, we were able to prove that
the gem box gives us a small, weak initial production of a gene.
And?
If we add the gen R, we get an induction and get more of the gene.
Fantastic.
Yeah, so we would certainly like to thank our sponsors, and thank you all for listening, and please, if you switch the slide, this is the whole team, so give them an applause.
Yeah.
Thank you so much, Kira and Nils, for this interactive presentation. Very nice. Any questions for Kira and Nils and the iGEM team? No, everything was crystal clear. Just one question over there.
What bacteria did you use? Did it originate in Yersinia or was it a E. coli bacteria?
The bactericin came from a relative to Yersinia enterocolitica called Yersinia fredriksen, which it uses as a target against Yersinia enterocolitica natively in nature.
Any other questions? If not, I will hand over a couple of books in Swedish this time about public speaking. So thank you very much.
Thank you.